Hi dear students, welcome back to Gene Interactions, and today we are going to concentrate on the non-allelic interactions which have been carried out in various organisms. As we had previously discussed, the allelic and non-allelic interactions are being found to be different. The interactions involving non-allelic genes which are located in the same chromosome or a different chromosome to express a single character come under the category of non-allelic interactions, and usually they express ratios, genotypic as well as phenotypic ratios, unlike the Mendelian ratios which were being obtained by Gregor Mendel in his experiments. And we can find that the non-allelic interactions can include various factors which will either complement or supplement each other, and they will produce a new phenotype. And based upon this, different strategies of genes are being evolved, like complementary genes, supplementary genes, inhibiting genes, duplicating genes, as well as the modifying genes, etc. So we can usually categorize the non-allelic interaction-based genes into various forms. And now we're going to talk about these genes in details and what are the different categories of them. So let's just explore what is it. Firstly, I would like to talk about the term epistasis or the inhibitory genes. Usually, we have come across a concept that there is the occurrence of either dominance or a recessive allele, and usually we find that the dominant allele will mask or obscure the effect which is being contributed by the recessive allele. Now, for example, if you have a tall in the pea plant, if you have a tall allele for the tall plant, the dominant that is dominant over the recessive allele small t, which codes for dwarfism. And so, tall and dwarf, the tall will inhibit the dwarf. So that is a case of dominant and recessive nature of alleles. But sometimes you can see that when different genes, which are not alleles, affect the same character in such a way that the expression of one masks or inhibits or suppresses the expression of the other gene, we call it as epistasis. That is, usually in the case of allelic interactions, what happens? We mainly consider those genes or the allele different forms of a particular gene. They are inhibiting or masking each other. That is the case of dominant or recessive. But sometimes you can see need not be the alleles of the same gene, but one gene is influencing the expression of another gene even when it is non-allelic in nature. Such a situation is referred to as epistasis. And usually, the gene that suppresses the expression of the other is considered to be as epistatic, and the gene whose expression is suppressed is referred to as a hypostatic. Now, usually, since these epistatic genes are inhibiting the expression of other genes, we call it as inhibiting genes also. And the term epistatic was first used in 1909 by Bateson. Now, this was a common concept of epistasis which we have come across from years. But now the concept of epistasis is being modified to a broader extent. Epistasis can occur in various scenarios. Either whenever two or more loci or of a particular gene interact to create a new phenotypes, or when alleles of one locus mask the effect of alleles at one or more other loci, and even whenever an allele of one loci modifies the effects of alleles at one or more other loci. So at different cases, epistasis can happen. And it is not only a case of inhibition, which is being considered when epistasis is being considered right now. And we say that it is an interaction at the phenotypic level of organization, And the genes that have been involved in a specific epistatic interaction may still show independent assortment at the genotypic level. And in such cases, the phenotypic ratios may be deviated from those expected with independent assortment. So, in this case, what I need to specify over here is previously we had thought that all the non-allelic interactions, okay, non-allelic interactions where a gene is being inhibited or masked by a non-allelic uh, version of a different gene, that was referred to as epistasis. Now here we can also have another case, not only inhibition but modification or altering of the effect of that particular gene is also coming under epistasis. So just keep that in your mind rather than telling that when a gene is, when the different genes which are not alleles affect the same character in such a way that the expression of one masks, inhibits or suppresses. Instead of that, you need to say it is when it masks, inhibits, modifies or suppresses the expression of the other gene, then we consider under epistasis. So based upon this principle, all the other non-allelic interactions, we have mentioned here about different non-allelic interactions, complementary genes, supplementary genes, inhibiting genes, duplicate genes, modifying genes, etc. All these non-allelic interactions will come into a type of epistasis which are being done in different ways. Previously, we used to talk about them as simply as complementary genes, supplementary genes, inhibiting genes, duplicating genes, and modifying genes, etc. But right now, the concept is that the epistasis uh, is a common term where non-allelic interactions will either inhibit, okay, or modify the non-allelic versions of a particular gene will inhibit, suppress or modify the interactions of another gene. That is referred to as epistasis. It can be by the effect of complementary action, or it can be by the action of supplementary action, or it could be by the action of inhibiting action, or by duplicates, as well as by the modifying genes. So the term epistasis would encompass all these concepts together, but basically they are being found to be considered to be as non-allelic genes which are located on the same or the different chromosome, and they influence the expression of a different character. So if you're going to consider epistasis right now, you can also divide them into two different ways. One is a dominant epistasis and the second one is recessive epistasis. As I mentioned earlier, there are some genes which will modify or inhibit the expression of another gene. That gene is referred to as and the one which modifies or expresses or modifies or inhibits the expression is referred to as epistatic. Now, if the epistatic gene is dominant in nature, then it is considered to as dominant epistasis. And if the gene which is inhibiting the expression of the other gene is a recessive gene, then it is considered to be as recessive epistasis. So, if a dominant gene masks the expression of an other non-allelic dominant gene, then we refer it to as dominant epistasis. When you come across with different examples, you will be more clear with it. And if a recessive gene masks the expression of a dominant non-allelic gene, that is referred to as a recessive epistasis. And the gene which is getting suppressed or modified here is referred to as a hypostatic gene. As I guess you remember this over here. The gene whose expression is suppressed is referred to as a hypostatic gene. So, as I mentioned earlier, epistatic interactions are of different types. It can be dominant epistasis, it can be recessive epistasis, or it can be duplicate recessive genes. That is, two genes, the recessive genes which are when they are being duplicated, they will give a action of an epistatic interaction, and they could also serve as complementary genes. and duplicate dominant genes are there, as well as duplicate genes with cumulative effect and dominant recessive interactions. Now you might think what is supplementary action, what is complementary action, and all that. We will be dealing that in the coming classes. So uh, based upon this, you can see that unlike the normal Mendelian ratios, as I mentioned here, different types of epistasis will have different types of interactions, or different ratios will be formed. Now, if you have a dominant epistasis, you will have a 12 is to 3 is to 1 ratio. And sometimes in recessive epistasis, you have 9 is to 3 is to 4, like that. Depending upon the type of epistatic interactions which have been happening, the ratios will be found to be different. But you can always see that in epistasis, less than 4 phenotypes will appear in the F2 generation. I hope so much is clear to you. We will be talking about the more details in the next session. Thank you for now.